So, we start back with diagram of the inner ear where I had explained to you the various parts, the cut section showing the cochlea. Now, the spiral of the cochlea has got it's got a circular part which is what is actually going around up to the apex of the cochlea. So, this is the apex. Now, if you look at a different diagram, there are several important things which need to be made out. So, one is this is the helical part which is the cochlea, this one unwound actually. So, then that is unwound because to have a sense that it is the same membrane and cavity. Now, the cochlea is divided into three sort of cavities, in the sense that it is called we will change the nomenclature subsequently. So, there is something called a Scala vestibuli, then there is something called Scala tympani. Now, the logic is that the oval window opens into this part of the cavity, and then there is this round window which is which opens into this part of the cavity. I put the directions like this. Now, I have spoken about the direction in which sound is transmitted. So, it is through the tympanic membrane, through the malleus, through the incus, and through the stapes. Now, when sound is transmitted, there is, I told you about amplification here. So, amplification is between the oval window and the sound is transmitted through this thing called as the Scala vestibuli. So, this is it is a 3D cut section. So, please do note that this is a 3D cut section. So, Scala vestibuli, and it goes through the Scala vestibuli up to the apex where it continues on the other side. So, that is why I meant by it being one single cavity, but it is called different Scala tympani. So, through there it goes way back over here and impinges onto the round window. So, if we map the pathway, it is steps, oval window to Scala vestibuli, goes across the helicotrema, which is the apex of the cochlea. From there it's actually a continuous cavity. So, that is what is important to be remembered. So, Scala tympani, and finally it gets dissipated into the round window. So, so that is the sort of pathway. It is a very complex structure. If you recollect the imaging which I showed you, I showed how big the size of the ear is and how small these structures are, and it is within these structures these smaller microstructures exist. Now, this is from the point of view of the sound wave, how the sound wave actually traverses through the inner ear, and so far I have not spoken anything about the sensor mechanisms. So, the entire sensor mechanism is present within the center of this structure called as the Scala media, which is this part over here. So, the Scala media is this area, and that has that's the important place where things happen. So, we progress on to the next video. So, next picture, and we will look at in greater detail of the cut section. So, scala vestibuli, so we know that this is in terms of the oval window, and this goes out, the sound is going out through the round window. It is not going out, it is just that there is a membrane and the sound which is transmitted across this fluid filled cavity, it just it gets dissipated sort of over here. Now, the central part is sort of sealed out excepting for this blue stuff which is seen over here. So, you have a very fairly rigid membrane over here which is called Reisner's membrane, and then there is bone over here, excepting for this membrane over here, which is sort of acting like a diaphragm. So, the basilar membrane is a membranous structure. This is a cut section, remember. So, this is part of the helix of the cochlea. Within the helix of the cochlea is this set of structures. So there are other features which are there. So, this the fluid which is over here is called perilymph, and this area would be the endolymph. So, endolymph, perilymph, perilymph is on the outside, endolymph has, endo is inside. So, endo is within these structures, and that is what is seen. There is also this funny structure called tectorial membrane, whereas the Reisner's membrane is this. This is called tectorial membrane. Now, all these are parts of this structure, but the important thing is called as organ of corti, which is these things over here. You will see it in the next in greater detail. So, this whole thing over here, from here onwards up to this part, is the organ of corti. I think it even includes the tectorial membrane. So, that whole thing is the organ of corti, and that is where the sensors are actually present. Now, they are called hair cells. They are called hair cells because they have hair. The tallest one is called kinetocilium. So this is a hair yeah, I did make it elaborate because each of, so we will jog back into the ear. So, we started with the external ear, which is this auricle which is over here, and then you have the tympanic membrane on air on both sides, goes through a set of bones, impinges on something called as the oval window, goes through the cochlea. So, cochlea, it is a two channel system actually. So, there is a central channel and then there is a peripheral channel. So, the peripheral channel goes all the way up to the apex where it meets with the other channel which is in continuity, and then that channel comes back over here as the round window. The round window is actually not connected to anything, it is just connected to air over here in the eustachian tube. Closer part of the closer section of the inner ear, oval window, round window, sound comes through the oval window, goes all over here, from here and comes up to the base, then takes another turn over there and then goes back over here and back to the base and comes back to the round window. So, the fluid through which it goes is the perilymph, the fluid through which it does not go through is the endolymph, Now, cut section of that showing the arrangement of the structure. So, the central chamber so as to say contains something called as the organ of corti, and then there is this blue line which is continuing all the way, separating this chamber from the below chamber here, which is the scala tympani, and that is extending all the way from the base up to the apex. So, that is what is shown here. So, another cut section in which you can clearly see the basilar membrane. You can notice that these cells are situated on this basilar membrane. So, the hair cells, which is written over here, this is the hair cell, several other kinds of supporting cells which serve several purposes. One is to stabilize these cells literally, in the sense that it mechanically stabilizes the cells. They also provide synapses, synapses which go specifically to the spiral ganglion, and this is the spiral ganglion. So, we go look into the organ of corti and the structure, how it is arranged. So, to give context, so this is the scala vestibuli, and then this is the scala tympani. So, we are in the middle part of the this is the middle part of that cochlea. So, this is to give context again, this is basilar membrane. On top of the basilar membrane is the cells. So, we start with the most important cell. This is the this is something called as the inner hair cell. The inner hair cell, as you can make out here, several synaptic connections, and these synaptic connections go on to the ganglia. Now, this is inner hair cell because it is towards the center. Towards the center of the cochlea. So, inner hair cell, then you got corresponding outer hair cells. So, outer hair cells are these brown things, and there are several of them. So, this is one single row. So, this one is inner hair cell. There are multitudes of outer hair cells, a total of I think 16,000, of which about 2,000 are inner hair cells. The remaining 14,000 is all outer hair cells. So, there are so many of them. So, these U-shaped stuff which you see over here are the cilia. Now, this cilia in turn is embedded within the embedded within the tectorial membrane. So, tectorial membrane is gelatinous. So, these are the players. So, we have introduced the players, where the players are situated, where the players stay, how the players are connected with the various parts, with the cochlear ganglion, and that is how the arrangement is made. A little bit more about this this diagram. I think I will be using this diagram later on also. So, you have got outer hair cells which are on the outside, you have got inner hair cells which are over here. Now, each inner hair cell you can make out as making multiple synapses. So, multiple synapses go to multiple cells within the ganglion, and from there the information goes back into the brain. I use the term brain, which is the cochlear nucleus, the olivary nucleus, the vestibular nucleus, to the inferior colliculus, to the primary auditory area. So, all this afferent is towards the brain, afferent is towards the brain. So, these are the signals which come. Incidentally, these are not these are again sensor actuator systems. It is a common theme. So, we had looked at this in our class on muscle cells and muscle architecture and muscle reflexes, where I used these concepts. So, these are again examples of sensor actuator systems in which the sensors are not just dumb systems of acquiring data, they are the sensors themselves are modulatable, in the sense that you can change your gain of the sensor. So, so the gain can be changed based upon whatever the requirement is, and then we see how the gain is changed by the mechanism. So, that is the actuator part of it. So, actuators can change gain. So, gain can be changed. So, this is the set of these are the this is how it is. Now, so we now understand certain things, and we understand how the fluid dynamics is fluid dynamics to some extent. So, now I can go to the box notation for the fluid dynamics. So, if we look at how things are, we have the Scala vestibuli, and then this is the round window, oval window. So, this thing continues up there, and now what actually separates yeah, so this is sealed. Now, what actually separates this these two things is the basilar membrane. And that is a membrane. So, that is a key thing. So, what membrane can do is can oscillate. So, we have started at very gross anatomical structures, and then we come down to this idea that it is basically a sealed cavity. So, sound comes from here, goes out sort of from here, and in the bargain what happens is the basilar membrane does this vibration, and this vibration is what is getting transmitted on to the cells here, and that sort of is going on to the nucleus. So, that is where the nerve signals are coming, nerves, and you got electrical signals. So, there are lots of membranes here. So, reiterating what I have earlier told. So, if we look at mechanism, so tympanic membrane to oval window to basilar membrane. So, if you look at what is done over here, it is amplification. So, then oval window to basilar membrane is also there is something else which is happening over here. So, what is exactly the basilar membrane doing? So, some properties of the basilar membrane are interesting. So, again we draw the whole we have unfolded so that to give a better idea of the basilar membrane. So, basilar membrane basically tapers. I have exaggerated the taper. So, this is something called as the helicotrema. now this whole thing is the basilar membrane. I think it is 33 millimeter in length. 33 millimeter is about 3 centimeters. Yes, 3 centimeters. So 33 millimeter in length. It has a length which is varying. So you have L1, then you have got L2. So L1 greater than L2. Now apart from this difference in length, what is important is its response to, it is a non-linear system. I have to explain both the non-linear and the system part of it. So what actually it does is, it's like this, and it sort of resonates to two waves. Now where did this wave come from? The wave came from the Scala vestibuli, which is coming from the oval window, which is coming from the stapes, and then from the tympanic membrane. Okay. So nonlinear system, so it just it sort of resonates. Now I use the term resonates instead of something else for a specific reason. So the entire membrane has differential response to differential frequencies. So frequency change, frequency dependent oscillation. So how is it? So it is you have it something like 20 hertz and 20 kilohertz. So 20 hertz, there is maximum oscillation in this part of the membrane. So this is the basilar membrane. So the apex of the basilar membrane vibrates with low frequency. Then I need colors. And high frequency, that is 20 kilohertz, the base vibrates. So there is a gradient. Gradient of sensitivity to frequency, input frequency. Input frequency is sorry, I think I shouldn't use the confusing terminologies. So the membrane is essentially split into various components. So I have just shown it as 20 hertz and so big basilar membrane. The basilar membrane deserves respect, and I will shortly tell you why it is so. In general, it is experimentally found that you can divide this into 3 components. So, 22 sorry 200 hertz, 200 to 2000 hertz, 2000 to 20,000 hertz. So, that is how it is graded. So, what I mean is when you have frequency in the range of 20 to 200 hertz, there is maximal displacement over here and no displacement over here when it is 20 to 200, and when it is 200 to 2000 hertz, you have 200. I think I should have marked it with a different color. So, you have maximum displacement over here, minimum displacement over here. And if we look at the more colors, if you look at the 2000 to 20,000 kilohertz, maximum displacement over here, no displacement in the rest. So, the basilar membrane is selective to frequency, and different frequencies causes amplitude changes only in selected parts of the membrane. So, that is the idea of saying system. So, it is just not a membrane, it is a system. So, the system is got a material properties which ensure that it is material properties. So, it material properties based upon one maybe diameter, and of course, I do not know the protein structure of this stuff, but the diameter, it is no, it is this, it is this thing, it is this thing, and that causes this membrane to oscillate at different frequencies. So, this is a concept which I need to explain in lot more detail, and so I will take lots more time to explain. Very important concept, and interestingly it is there are, I think, somebody, at least some people in the audience by now, there is an aha moment once I spoke that this selective stimulation. Now, so that is one part of the system. So, one part of the system story. So, system story of the basilar membrane. So, basilar membrane again 20 hertz, 20 kilohertz. So, if you look at, so we plot frequency. I do not know, I should call it resonating frequency. So, the part of the membrane resonates to a particular frequency, that is the idea. So, resonating frequency, and then you got the basilar membrane. So, apex, base. So, we start at 20 hertz to 20,000 hertz. So, if you would plot that, you would notice that it is I think the curvature is a log. So, that is a log curve. So, that is one more reason why I call it as a system instead of just a membrane. So, so the membrane what you call gets excited by very different frequencies at very different parts of the membrane. So, that is the part of the story to which we have come down. A couple more people would have the aha moment at this time, because there is a log curve and then there is a resonating frequency, and then there is a base in the apex. So, we go to the next part of the story.